Welcome to this explainer. Whenever we think about the biological cell, it's so easy to picture it as this passive, static little blob, just peacefully floating there under a microscope. But today, we are going to completely reframe that perspective. I want you to start imagining the cell not as a simple, inert water balloon, but as a bustling, high-speed, biorobotic metropolis. It's an intricate city with its own massive highways, dedicated manufacturing plants, and complex communication networks, all operating at a scale and a speed that is, frankly, almost impossible for the human mind to comprehend. So let's dive right into this explainer with a pretty staggering number, 100. Inside your cells, at this exact second, there are microscopic molecular motor proteins called kinesin. They're physically sprinting along cellular highways, pulling these massive cargo structures behind them, and they are taking exactly 100 steps per second. To put the sheer importance of that into perspective, if these tiny biorobotic engines were to suddenly stop moving for just 3 seconds, you would drop dead. That is the sheer high-stakes velocity operating within the engine room of life.
To make sense of this immense microscopic universe, we've mapped out our explainer into 5 structured systems. We're going to cover the dense cytoplasmic metropolis, organelle assembly and transport, power plants and waste management, how cells communicate, and finally, the cell cycle and cancer. Let's start with the city layout itself. Section 1, the dense cytoplasmic metropolis. Now, this is going to completely shatter that high school textbook drawing we're also used to. We typically see a diagram showing the cell's interior as a clear, empty glass of water with just a few organelles floating around peacefully. But the biological reality? It's drastically different. The cytosol isn't thin water at all. Imagine trying to run through a swimming pool filled with thick, viscous honey instead of water. That's the environment inside the cell. It creates this ultra-dense, continuously jiggling molecular traffic jam that biologists call macromolecular crowding. That incredible density is caused by packing a staggering 300 grams of macromolecular solutes into a single liter of space. Because everything is packed so tightly, molecules don't just gently float by one another. No, they are forced to collide millions of times faster than they ever would in dilute water. This massive molecular traffic jam is so intensely chaotic that it actually boosts metabolic enzyme kinetics by up to 1,000-fold. The chaos is exactly what makes the chemistry of life possible.
Okay, now that we understand the dense streets of the cytosol, let's look at the infrastructure. Section 2, organelle assembly and transport.
To hold this dense city together, the cell relies on the cytoskeleton, which is a physical framework made up of 3 distinct components. First, you have the 25-nanometer microtubules. These are hollow, rigid cylinders acting as literal two-way highways for those kinesin and dynin motor engines we talked about earlier. Then you have the 7-nanometer microfilaments, acting a lot like dynamic muscle fibers that allow for things like cell crawling. And finally, the intermediate filaments. These act as high-tensile braided steel cables. They don't help with transport at all. Their whole job is just to absorb mechanical tension so your cells don't collapse or tear apart under pressure. Deep inside this complex web of highways sits the nucleus, the central command vault. What is truly mind-blowing here is the packaging problem the cell has to solve. It has to pack 2 whole meters of linear DNA into a microscopic 6-micrometer space. I mean, if you just shoved it in there, it would tangle and snap instantly. So the cell wraps this incredibly long genetic thread around basic proteins called histone spools, perfectly coiling and looping it over and over again so the code remains safe, highly organized, and ready to be read at a moment's notice. And when that heavily guarded genetic code actually gives the order to build a protein, it follows a brilliantly efficient assembly line known as the secretory pathway, which was mapped out by Nobel laureate George Palade. First, synthesis begins on the ribosomes bound to the rough endoplasmic reticulum. Then, the newly made protein is transported via Koppai vesicles. Next, it arrived at the receiving cis-face of the Golgi apparatus for crucial processing. After that, it moves over to the trans-Golgi shipping dock where it gets a specific molecular tag. Finally, it undergoes secretion right at the plasma membrane. It's just an ultra-efficient, flawless multi-step factory. Of course, every massive manufacturing hub generates trash and requires enormous amounts of energy, which brings us to Section 3, power plants and waste management. When it comes to waste management, your cells do something absolutely incredible. They literally eat their own damaged parts to survive. This process is called autophagy, which translates directly to self-eating. When a piece of cellular machinery naturally wears out, the cell drops it into lysosomes. Think of lysosomes as cellular garbage disposals that maintain an intensely dangerous acidic pH of around 4.5. That harsh acid and specialized enzymes chop up this molecular garbage, digesting worn-out organelles and turning them right back into raw amino acids that the cell can efficiently recycle. Now, to power all of this incredible digestion, movement, and manufacturing, your mitochondria run one of the most stunning mechanical engines in all of biology. As protons are pumped across the mitochondrial membrane, they flow back through a microscopic rotary engine called the ATP synthase. This is a literal nanoturbine embedded right inside you, mechanically spinning at up to 7,800 revolutions per minute. It spins with such intense velocity to smash molecules together, generating the ATP energy that ultimately keeps your heart beating and your lungs breathing.
While animal cells rely primarily on those high-speed mitochondria for their needs, using small, temporary vacuoles for basic storage, plant cells operate a bit differently. They pair their solar-farming chloroplasts with a massive central vacuole. This central vault can take up to 90% of the entire cell volume. It draws in water to act like a massive internal hydraulic jack. This generates immense turgor pressure from the inside out, which is exactly what keeps the non-woody stems of plants standing perfectly upright against gravity.
But remember, a single cell doesn't just exist in a vacuum. It has to coordinate with billions of others. That brings us to section 4, how cells communicate.
Cells effectively use 4 primary radio frequencies to talk to each other. They use endocrine signaling for long-distance hormone drops through the bloodstream, paracrine for local neighborhood chats, autocrine to talk to themselves, and juxtacrine for direct, face-to-face contact. Once a signal is received at a cellular receptor, it fires off what are called second messengers inside the receiving cell. Molecules like cyclic AMP act as these messengers, and they can take a single, lonely signal, like a tiny drop of adrenaline, and amplify it a staggering 100 million-fold in just seconds through a massive signal transduction cascade.
And when these cells need to physically link up and share information directly, they lock together using 3 specialized junctions. Tight junctions act exactly like waterproof Ziploc bags in places like your stomach lining, ensuring harsh acid doesn't leak out into your body. Desmosomes act as heavy-duty mechanical rivets for tissues that are constantly under high-stretching stress, like your skin. And gap junctions form literal open communication tunnels. This is absolutely vital because it allows ions to pass seamlessly between neighboring cells, ensuring things like your heart muscle cells all fire and beat in perfect, uninterrupted electrical rhythm.
All of this infrastructure builds to our final highest-stakes topic, Section 5, the cell cycle and cancer. In order for you to grow and heal, your cells must actively divide. To do this safely, they navigate a flawless 4-stage timeline called the cell cycle. The cell actually spends about 90% of its life in a slow, careful stage called interphase. It moves deliberately through G1 for growth, then an S phase where it perfectly and meticulously copies its entire 2-meter genetic genome, and then G2 for final preparations. All of this slow, highly regulated preparation leads up to the M phase. The M phase is a rapid, violent, 1-hour frenzy of mitosis where the chromosomes perfectly align and are instantly pulled apart to form 2 completely new cells.
Because this division process is so incredibly dangerous, the cell has rigid internal security checkpoints. If a cell detects a critical error, like irreversible DNA damage during the cycle, it triggers a fascinating and deeply complex self-destruct sequence known as apoptosis. This isn't some messy, chaotic explosion. It is highly programmed cell suicide. The cell neatly chops up its own DNA, cleanly shrinking and dismantling itself into tiny, membrane-bound fragments that can be quietly scavenged by your immune system without causing any tissue inflammation. It literally sacrifices itself to save the rest of the city.
That honorable title, the Guardian of the Genome, belongs to P53, the ultimate tumor suppressor gene. It acts as the strict molecular security guard stationed at those cell cycle checkpoints. When it detects damaged DNA, P53 literally slams the brakes on cell division until the damage can be repaired. But here is why this matters to every single one of us. When the P53 gene itself mutates and those security checkpoints completely fail, the cell simply ignores the stop signs and begins dividing out of control. That catastrophic, unregulated division is exactly what we call cancer.
So, as we wrap up this explainer, I want to leave you with a final thought. The next time you look in a mirror, don't just see a single organism looking back at you. Remember the trillions of bustling, high-speed biorobotic metropolises, complete with walking motor proteins, specialized acid waste centers, and 7,800 RPM nanoturbines, all working in perfect, perilous harmony right this very second just to keep you alive. Stay curious and keep exploring the masterpiece inside you.